Thank you so much for the kind introduction. It's my great pleasure to tell you today about a recent project where we have developed a novel strategy for measuring gene expression on the level of the whole genome and also with spatial resolution. Before I go there, let me start with a very big question that I think has fascinated mankind for centuries. It's the question of how a single cell, a fertilized egg, can turn into a fully grown organism consisting of millions of different cells that are arranged in a complex spatial body plan. This cartoon here shows a zebrafish, because that's the model organism that we happen to work with. But this question of how patterns emerge applies equally to all other species, including humans. And I think the emergence of spatial patterns at many different cell types is really a most striking phenomenon, in particular if we consider that in our bodies we have many different cell types, hundreds of them, for instance, the cells in your muscles, in your brain, in your skin, etc. Now, the construction plan for our bodies is in the genome, in the DNA. Work in the last decades has clearly shown that. Now, if you take those two last points together, I think a question emerges somewhat naturally. If every cell has the same DNA, how is it possible that so many different cell types can be created and generated? The answer is that it depends on which part of the genome is active. Let's take a step back and let's look at what's known as the central dogma of molecular biology. As I said, the genome is the construction plan of our cells. The DNA contains the genes, which are then transcribed as RNA and later translated into protein. Now, these processes of transcription and translation are very heavily regulated. And each cell transcribes only a subset of its overall around 30,000 different genes. And it's this subset of expressed genes that determines the identity, the type, and the function of a cell. So knowing where genes are expressed is a very important piece of information if we want to understand how patterns form in development and how processes get deregulated in disease. So how can we measure gene expression, transcription of RNA? The traditional classical approach is based on microscopy. This here is a cartoon sketch of a zebrafish at about an age of 6 hours. You see the spherical yolk sac that provides nutrients in the middle, and the embryo proper in gray is sitting on top of it, sort of like a cap. So to measure gene expression by microscopy, you can use different approaches, in situ hybridization or GFP fluorescence. Here's an example where we're looking at one particular gene called goosecoid or GSC, which is expressed only here at the right-hand side. And as you can see in these microscopy images, we can measure these spatial patterns with good spatial resolution. That's very good, but it's also difficult to measure all 30,000 genes at the same time, because we're limited to studying just one gene at one time. So this means getting all 30,000 of them would be very time-consuming. It would also require a lot of samples, 30,000 embryos in this case. Now, over the last couple of years, a different orthogonal approach has appeared called transcriptomics, where you measure the expression of the whole genome at the same time by sequencing the RNA. To do that, you take your samples, extract the RNA, and then in a test tube, determine the nucleic acid sequence of the different RNA molecules. So for instance, for this molecule here, you'd read AG, CT, GT, etc. So this sequence information for each molecule tells you which gene it is derived from, and you can also find how many copies you have. So this is very powerful because it gives you all the genes at the same time, but without any spatial information, because you do this in a test tube. So this brings us to a fundamental trade-off between genome-wide techniques and spatial resolution. Right, the sequencing-based technologies are genome-wide but don't have spatial resolution. The spatially resolved microscopy-based techniques are limited to just one or a few genes at a time. We wanted to solve this dilemma, have the best of both worlds, and do spatially resolved transcriptomics. Now, having such a technique would be very powerful in many fields of biomedical research. If you think about cancer research or cardiovascular biology or regenerative medicine. But keeping with the introduction based on developmental biology, I think it's also very easy to see why in development it's important to know where a gene is expressed. If you know where a gene is expressed, it's typically very good information about its function. Imagine you find a novel, uncharacterized gene that is expressed only in the developing heart, but nowhere else, and it's very likely that this gene, in one way or the other, is involved in heart formation or heart function. So let me show you a short video as an illustration of how this technique works. I'll show you a 3D image of a zebrafish embryo, and the video will move from left to right through the individual planes of the image. You see all the different cells of the zebrafish embryo in black and white, and now the expression domain of one gene, goosecoid, appears here in green and disappears again as we move to the side. So again, this is just one gene. We have thousands of additional genes from the same samples. And here's how it works. It's actually a very simple idea. We take our sample of interest, we cut it into small pieces, and we sequence the RNA from the different pieces individually. I call this approach tomoseq, inspired by the ancient Greek word tomos, which means slice or section. So this here is a sketch of a zebrafish embryo at a slightly later stage. Again, you have the spherical yolk sac in the middle, and the embryo proper in gray is sort of wrapped around it. Now we cut the zebrafish embryo into about 100 thin slices using a cryotome, an instrument for making tissue sections that you find in every clinic or biomedical research institute. Instead of looking at these slices under a microscope, we put them into individual test tubes. We extract the RNA from each of them, and then we do a couple molecular biology tricks, like we add a different molecular barcode to each of the sections, which then allows us to pool them all into one tube. Then we amplify them in a very controlled manner, and then we can subject these libraries to sequencing. So here's an example. In the simple example, we took such a zebrafish embryo, we removed the yolk, and we straightened it, and then we sectioned it into 100 slices from the head to the tail. Then we can create traces like the ones shown here. So we find that 6-3A here is expressed only in the head. We find MIL7 in the heart, the gene MESP-AB in the nuosomite, that's a transient embryonic structure, or TBOX16 in the tip of the tail. Overall, in the sample, we detect more than 20,000 different genes with very good signal-to-noise, so you see that the spurious background level outside the expression zone is almost zero. So this looks very promising, but well, this was one-dimensional information, and the video that I showed you initially, and what I promised you, was in 3 dimensions. So how do we move from 1D to 3D? It's again a simple solution. Now we take 3 different embryos, and we section them along different axes, along the 3 main body axes. So from left to right, ventral to dorsal, and from the head to the tail. That way, we measure projections of gene expression along the 3 main body axes. And then, based on these 3 1D data sets, we reconstruct a composite 3D image. This is inspired by optical tomography techniques like computed tomography, where you also take images under different angles and then reconstruct an image. Here, the mathematics is a bit different. I'd be happy to discuss that later if anyone is interested. So at first, let's look at how this works. Let's again look at a younger embryo at shield stage, where the embryo looks like a cap on top of the yolk. So this is clearly a structure that you couldn't separate from the yolk and straighten without completely destroying and distorting the structure. So we need a 3D approach. Again, we're looking at the gene goosecoid expressed here. In each of the 3 one-dimensional data sets, we find a clear peak of expression. Then, as another ingredient, we measure the shape of the embryo in 3D with light sheet microscopy, and then using mathematical image reconstruction, we can put the individual pieces together. And this is indeed the exact same video that I showed you initially. So this is very nice because it gives us a genome-wide compendium or an atlas of gene expression. But I think what's maybe most powerful about this is that we have a data set that we can immediately search for any spatial expression pattern that we're interested in. For instance, we can rank all genes by similarity to a known reference gene involved in a certain function. We did this here by asking, what are the 15 genes that are most similar in expression pattern to goosecoid, the example that I showed you? And when I play this video, you'll see in a moment that when the expression of goosecoid comes on, they all beautifully light up in unison and also disappear again together as we move to the side. Now this shows us two things. First, that we found a way to quantify similarity of these expression patterns. And secondly, that there are many genes that have similar spatial expression patterns. These are just the top 15. As we go down that list until around position 100 or so, genes are still very similar to the reference. Among those genes are many known genes. That's good because that's validation of the approach, but we also find many novel genes, and those are the ones that are, of course, biologically most interesting. Now, to be sure that what we measure here is real and not a false positive result, we decided to validate a few of those novel genes with traditional in situ microscopy methods. So here at the top, again, you see the reference goosecoid in the 3D reconstruction TomoSeq image, and this here is microscopy data, which, as you can see, is identical. And now here are 6 novel genes that we took out of the list. If you compare these images, these are maximum projections taken from the 3 main body axes. I think we can agree that these novel genes are all highly similar to the reference data set, suggesting that this allows us to reliably identify novel genes with spatially restricted expression patterns. Now, I'd like to say that all of these genes look slightly different. For instance, Ripley1 here is expressed a bit more broadly along the left-right axis. UNX41 has a very narrow expression pattern, but overall this performs very well. So let me summarize. I've shown you that we've developed a novel method for spatially resolved transcriptomics in 3D, and we've generated a large resource of expression patterns in the zebrafish. What I think this is most important for is as a gene discovery tool that we can use to understand how development performs in healthy individuals and how processes get deregulated in disease. In current work, for which we have received an ERC starting grant, we're now expanding this approach to also include information about the history of cells, and then we want to use these approaches to study how the heart reacts to perturbations and can correct these perturbations. So with this, I'd like to end, and I'd like to thank the people in my group who've made all of this possible, and I'd like to thank you for your attention. I'd be happy to take any questions.